you. Good afternoon, everyone. My name is Rupert Stacy, and I'll be presenting my thesis, Retained Genetic Diversity of Hippocampus Guttulatus in the face of demographic decline in Ria Formosa. But before I start talking about the specifics of the project, I'll introduce you with a little bit of preamble about the field my project is based in. This will lead nicely into the importance of the study, the hypothesis and aims, the methods, and then obviously the results and the discussion. Yeah. So this thesis primarily focused on genetic diversity. While we have studied other genetic elements, the primary concern was of genetic diversity and its loss in the subject of study. So the Convention on Biological Diversity was ratified by 196 parties and aims to conserve and protect diversity at three different levels. They are ecosystem diversity, species diversity, and genetic diversity. And the reason for focusing on genetic diversity, it is the basis for both of the forms. While I'm not trying to say that genetic diversity is the most important, I'm just trying to say it underpins both other forms. So what exactly do I mean by this? Well, genetic diversity is the raw material that natural selection acts on. It enables species to speciate and create diverse ecosystems like forests or coral reefs. It's important to have genetic variation as it enables both species and populations to adapt to different environments and pressures. Like I said, it's the raw material that is selected through the process of natural selection that enables the fittest, or should I say best adapted, to survive when faced with diseases or changing environment or predators, for example. And really importantly with genetic diversity is that when a population is reduced and it has lost genetic diversity, it's lost for good. As you can see in this graph, a population may recover in terms of both size or number, but if genetic diversity is lost, it's gone for good, except for two scenarios, which are if there's gene flow from another population, so long as there is another population, or from mutation, but this takes place at an evolutionary timescale. And populations that are greatly reduced and have lost genetic diversity can often suffer genetic bottlenecks, a famous example of which is the African cheetah, which was supposed to have gone through a genetic bottleneck around 10,000 years ago, which you can still see evidence for in the form of low genetic diversity. But the reason I mention all of this is because genetic diversity minimizes extinction risks, but also gives species and populations resilience, like I said. So having set the scene, I'll talk more about the project. I was looking at the long-snouted seahorse, Hippocampus guttulatus, and occurring in the Ria Formosa in Portugal. It was recorded as being having the highest densities on record in 2001, and then 2002, sorry, and then by 2008 it experienced a 94% decline. Although the cause of the decline is still maybe debatable, the primary factors for this are thought to be illegal fisheries and also habitat degradation. And although the population did recover by 2013 to similar levels as seen in 2001, the decline had prompted fears that the population may have lost genetic diversity or had caused a genetic bottleneck. Which brings me to my aims and hypothesis. So we used samples collected in both 2001 and 2013 to answer the following questions. So did the decline reported in 2008 cause a reduction in the genetic diversity of Hippocampus guttulatus in the Ria Formosa? Was the decline severe enough to have caused a genetic bottleneck? Had the decline resulted in the effective population size being reduced? And what effect had the decline had on the level of inbreeding in the population? So my job within the thesis was to analyze and interpret the data that was given to me. So after some preliminary processing, our final data set consisted of 8 microsatellite loci with 33 individuals in 2001 and 258 in 2013. And from there, we use a number of softwares. We used the diversity package in R to calculate various genetic diversity indices. We used INEST and BOTTLENECK to determine whether a genetic bottleneck had taken place. We also used INEST to calculate inbreeding coefficients using a Bayesian approach to account for null alleles, if there was any. We also used ML-relate to calculate the amount of relatedness in both years of samples. And lastly, we used NE estimator to calculate effective population size from levels of linkage disequilibrium. And you may have questions about sample sizes, and where possible, we used resampling or rarefaction techniques to standardize these findings so they could be directly compared. So moving on to the results. Firstly, the genetic diversity results. I should mention that these are the averages over the 8 loci. As you can see, they've all increased. The biggest increase is in the number of alleles, but it's far more informative to look at the allelic richness and private allelic richness here, simply because they've used rarefaction and aren't as biased to sample size. Similarly, both expected and observed heterozygosity has increased, very marginally maybe, and although they could have been biased from sample size, it's thought that around 30 individuals is needed to accurately capture the level of heterozygosity in a population. So they can be compared relatively well. And lastly, the inbreeding coefficient on the far right shows a decline. While I haven't included the results of the Bayesian approach to calculate the inbreeding coefficient, simply because the results from it didn't really change all that much. But one really interesting aspect of the Bayesian approach was that it gave us 95% confidence intervals, which did not include 0. So we can safely suggest that some mild to weak inbreeding was present in both years of samples. As for the decrease in the inbreeding coefficient, I'll come back to this shortly when I speak about it in the discussion. So next is the bottleneck test. We use 2 methods, heterozygote excess and one that looks at the distortion of allele frequencies, and neither of them suggested that a genetic bottleneck had taken place from 2013 as a result of the decline in 2008. The results of the relatedness, we found 4% of the samples in 2001 to be related as half-siblings, whereas in 2013, this percent doubled to 8%, and we even found a small number of full siblings. And lastly, the effective population size. So let me first explain the graph very quickly. On the x-axis, we have the years on both sides of the graph, and then on the y-axis, we have the effective population size in numbers. On the left, we have the full data set with 33 individuals in 2001 and 258 individuals in 2013. Whereas on the right, we have our resampled estimates, where we resampled 2001 and 2013, 30 individuals 1,000 times. So the black dots on this graph represent the lower bounds because our point estimates often included infinity, but reporting on the lower bounds are really informative in terms of levels of change. As you can see from the left graph, it looks like the effective population size has increased, albeit with overlapping confidence intervals, whereas on the right, there is very little change in the lower bounds. And these are the best estimates we can get for comparing these 2 years in terms of the difference in sample size, and they still show an increase in the point estimates represented by the red dots. And in both cases, the point estimates center around the 2,000 individual mark. So what does this all mean? Well, as we've seen, the decline in 2008 did not lead to a situation where the population in the Ria Formosa had lost genetic diversity and nor had it caused a genetic bottleneck. And why, you might ask? Well, it's possible that the 94% decline may not have been as big as the 94% may lead you to believe. Like, it's possible that the decline may have brought the population down to numbers comparable with other populations. As I said, the 2001 population was the highest on record. And there's a general rule of thumb that says between 500 and 1,000 individuals is needed to retain evolutionary potential, which is somewhat reliant on the level of genetic diversity that it contains. So given our results of an effective population size of 2,000, it's possible there was enough individuals to carry through genetic diversity throughout the decline. So yeah, 2001 and 2013 have similar levels. Alternatively, another explanation could be gene flow. So this image was taken from a study by Riquet last year, and it shows the populations of Hippocampus guttulatus around Europe. So I'd like you to focus on the blue area on the left. And it shows that the Iberian Peninsula the populations around the Iberian Peninsula are panmictic for a panel of 260 SNPs. Essentially, what this implies is there is gene flow or migration between the populations in this area. Another study in 2009 hints at a very similar situation, albeit this time with a smaller number of microsatellites. But what this basically means is the Ria Formosa is not a closed population, and that if genetic diversity is lost, individuals, likely as larvae, may disperse along the coastlines and enter the Ria Formosa and provide genetic diversity. One interesting aspect about Lucy Woodall's study in 2009 is that she did find a genetic bottleneck from samples that she took in 2004. Interestingly, the higher inbreeding coefficient in 2001 is likely to be a result of this previously found genetic bottleneck, and that the 2008 decline may have actually purged the population of slightly inbred individuals. Similarly, this also kind of explains the doubling in the relatedness. With fewer individuals post 2008, you've got a much higher likelihood of sampling related individuals due to the remaining individuals directly contributing to the recovery seen by 2013. So, in conclusion, the long-snouted seahorse was able to retain much of its genetic diversity through a 94% decline, possibly due to the sheer size of it, possibly through gene flow, possibly both. But I would recommend that future genetic studies may want to look at the number of migrants per generation, as this is supposed to be quite important, and to identify if there is any direction to the gene flow, if any is found, as protecting the sources of larvae could be a really interesting conservation approach. But in terms of seahorse conservation, I would recommend that the best course of action is to repair and restore degraded habitat, which is quite a lot here in the Ria, especially seagrasses, but also tackle the problem of illegal fisheries that is supposedly contributing to the decline. With enhanced enforcement and protection. So, and also considering the population is reported to have experienced another decline in 2008, I would suggest that continued genetic monitoring should be carried out, especially in terms of genetic diversity or inbreeding coefficients. Some references, and yeah, I would like to say a massive thank you to Professor Rita Castillo for really dedicated support, and Professor Jose Pedro Andrade for giving me the opportunity to use his data. Thank you all for listening. Now time for some